Okay, so welcome everybody to our second session on FCS Express in 2021. So we've had plenty of sessions in the past, but this is our second for 2021. I'm very happy to introduce today Sean Burke from DeNovo Software. He's been gracious enough to offer this training for us today on compensation and an introduction to high dimensional data reduction in FCS Express. And just a little bit about Sean. He is incredibly helpful. I know that I've reached out to him many times in the past and he's been able to help us out. But he's been with DeNovo Software for almost 11 years this year in 2021. And he's the director of research, marketing, and business development. And he has a plethora of knowledge. So I highly encourage anyone to reach out to Sean with any questions you might have. And just throughout today, as I pass along to Sean, if anyone has any questions, please feel free to use the Q&A function or the chat function. If you want to go ahead and ask any questions, and we'll be happy to go ahead and get those addressed. So with that, I pass it along to Sean and we're good to get started.
All right. Yeah, thanks, Kathy, and thanks everybody for joining us today. So today we're going to be going through some of the compensation tools in FCS Express and also giving folks a kind of introduction to high dimensional data reduction using tools like TISNE and VISNE and FlowSOM. And you might wonder, why do we kind of pull together these two topics? And really, in reality, compensation is a type of transformation on your data, just like TISNE and VISNE and other tools are transformation on your data too. And I also think, if folks are getting into some of the post-acquisition compensation in FCS Express, generally it's because they're working with kind of higher dimensional or higher parameter based data sets. So I thought it'd be a good kind of intersection between the two topics. And as for a little agenda for today, we're going to get started by looking at compensation with FCS Express. Essentially, what we're going to run through today is how to use the automatic compensation wizard in FCS Express. And I put a little topic here for what not to do. I know there's a lot of folks who like to do manual compensation of their data. I think it's a very dangerous thing. However, we do get requests and questions all the time about how do I move my data around on my plots after I've done some sort of compensation show. We'll show that to you. And again, if you are working with compensated data, right, if you're working with compensation, you find yourself, even if you're using other software to kind of move populations around on your plots using the compensation tools, I highly encourage you to reach out to the flow cytometry core facility. As long as you have good controls for your data, there's no reason that you'll need to be manually compensating data by eye. You're literally just moving things around the plots at that point. So again, you can reach out to Kathy, Rui, anybody at the core facility to kind of help you out if that is something you find yourself doing. Now, the second portion here, again, we're going to come in and not just, teach you about how to use TISNE and FCS Express. What I want to do is give folks a kind of overview of working what TISNE actually is and kind of a primer. Now, really importantly is going into some kind of common pitfalls that folks run into that they might not know about before they run TISNE or they run VISNE or FlowSOM and things like that. And I'm going to spend a lot of time talking about that because we often get questions at DeNovo Software, just as I'm sure many of our competitors do as well, where somebody writes in and they say, I ran a TISNE on things. The plots look different between different data files and all this kind of good stuff. They've already spent, 10 hours working on this stuff. And then we tell them, when you run a TISNE, you have to merge all the data files first. And they didn't know that. And then they have to go back and spend another, X amount of time to kind of work with that. So I'm going to go over a number of those things in that second portion of the talk. And hopefully, that will give folks a good starting place for working with the tools in FCS Express, which are actually very easy. So one place I like to start, with a lot of the talks I give is, why use FCS Express? In the past, folks have used things like cell phones and GPSs and cameras and calculators and computers. So they had lots of different tools for doing everything that they do in daily life. And that's all wrapped into a smartphone now. So that makes life very, very convenient. And kind of similarly, FCS Express has all the tools that other data analysis software has. We go above and beyond, of course, a lot of our competitors, but we can also integrate spreadsheets into the software. So when you move a gate, everything in your Excel like spreadsheet updates in real time. We integrate PowerPoint capability and exporting the PowerPoint in high resolution graphics. Kind of importantly for today, we wrap in Tisney, Visney, UMAP, FlowSum, a lot of the things you'd be doing in R, a lot of things you'd be doing in other software using plugins, all within the FCS Express interface. So you don't have to know how to program code. You don't have to know how to write R scripts. You don't have to figure out how to plug something into a piece of software to get out a result. And you're going to see, again, the interface for doing all of this in FCS Express is very straightforward, right? So that allows you to kind of focus on the biology and focus on the results that are going to be coming out rather than just figuring out how to use the darn tool. All right. So before we kind of start, I do want to remind everybody that everything we see today in FCS Express, you can do it on a Mac or PC. You can do that interchangeably. You can save something on a Mac and open it up on your PC. The site license at Memorial Sloan Kettering Cancer Center allows folks to install FCS Express on as many different computers as they want, right? So on a snow day like today, you can take your data from the lab. We're working on a PC, you can run it on your MacBook at home, bring it back to the lab, whenever you get dug out and get going. We are going to read data from every cytometer that's on the market, right? And that also includes all of the compensation support. So if you're acquiring it on Diva and you're doing compensation on Diva, so I call that, compensation at the time of acquisition, that's fine. SCS Express is going to read it in directly. If you decide that you want to change that and do post-acquisition compensation from the same data sets using controls, FCS Express is going to support that. And again, that's going to go for every cytometer, every kind of software that you're acquiring with on the market. So I'm going to kind of jump into that a little bit more. And again, what we're going to do is read compensation from any data files that you may have. Our auto compensation wizard in the software that we're going to take a look at in just a moment is going to calculate compensations from any single sane controls, and then you can use that downstream for whatever it is that you want, right? But it's important. You have to have good controls. If your controls are bad, the compensation wizard isn't going to do such a great job, right? And that's not just with FCS Express. That's with any software on the market. Every software package out there pretty much calculates compensation in the exact same way, and it's all based on providing the correct controls for your data. So slider bars for compensation are available, but they're really an antiquated and actually quite dangerous way of changing compensation, right? I'm going to focus on that in just a little bit. But if you're using slider bars, you are actually moving your data around in the plot without any sort of guidance, right? And that's a very important thing to note, right? That's how, you know, grants can be lost. That's how, papers can be returned to you. We want you to avoid that kind of stuff and just use proper controls. If you use the proper controls, it's going to work every single time, even if you don't use FCS Express. So I can't stress that enough. All right. So let's jump into working with compensation in FCS Express. We'll show you a little bit about how this works. All right. So I've got FCS Express open here, and I've got some data files that I want to compensate. Let's see, I think they're popping up on my other screen, hidden behind a bunch of Zoom windows. So let me go find out where that stuff went. Apologize. I've got a lot of windows open today, but I found the correct one here. All right. So essentially what we have in this data set is we have a all color control for a data file. And this was actually compensated at the time of acquisition. So if I wanted to pull this all color control into FCS Express, I'm not going to go over all the basics about loading data and stuff like that today, but I can drag it and drop it into the layout. And what you're going to see in the title is it's going to say compensated. right? And that means that some sort of compensation is applied to this. And if I come in here, just kind of quickly, I'll look at forward scatter versus side scatter. I'll create some sort of gate so I have something appropriate to look at here. And I'll drag this out and just look at, maybe CD4 versus CD8, right, or CD8A. I can see, this data looks like we'd probably expect, right? We have kind of populations straight up and down on the vertical, pretty much left and right, nicely compensated. So again, this is data that was compensated at the time of acquisition. If I go into the Tools tab, this is where we keep all of our compensation and mixing tools. All right. And if I expand any of the nodes that are here, if there's a compensation associated with the data file, whether it came from the time of acquisition or somewhere else, like I'll show you in a little bit, that entire matrix is going to be displayed. You'll be able to come in here and view this and visualize it. We have heat mapping to this sort of stuff, so you can kind of get an idea of what's high, what's low, and you can kind of keep track of all of that. Now, one of the unique functionalities in FCS Express, before we go in and compensate some of our own data, is that we can compare compensated to uncompensated data at any time, or compare multiple different compensations. Maybe you have data you want to look at uncompensated, you want to compare that to compensation at the time of acquisition, to post-acquisition compensation. We can do all that, and it's all controlled in the formatting of plots. So if I right-click on this plot and I choose Format, right? This is our kind of familiar formatting tool in the software. Well, in our overlays, which is just essentially telling us what's on that plot, we're able to come in here and select which compensation is being used. So if I set this plot on the bottom to none, right, we can see now this is what that data looks like uncompensated, and we can compare that very easily to compensation done at the time of acquisition. Right. So again, if you have multiple compensations, whatever it is you want to compare, keep in mind you can always kind of apply it to the plot. You can remove it from the plot. You can compare multiple plots. Any of the compensations that you are working with up here, you can drag and drop that onto a plot like I'm doing here, and that plot will then use that compensation. Right. So that's kind of some of the basic functionality for getting compensation applied, removing compensation from plots. Again, if you have compensation at the time of acquisition, it's going to load up automatically in there. Now, I'm going to show you, the most dangerous thing you can do with your data. I'm going to give you 20 seconds on this because you should not be doing it. However, I know a lot of people are. If you want to come in and you want to, move this data around without using your controls and, trying to compensate by eye, you can right click on a plot. You can choose the compensation that's applied to that plot or create a new one. And FCS Express will provide you with slider bars. Now, again, if you are using these slider bars like this, you are moving your data around the plots willy-nilly, right? There's no kind of rhyme or reason to where you're putting these things. That's impacting the data for this parameter, for other parameters that are involved in your data set, and you should not be doing it. And that's why I'm going to click Restore. Right, I'm going to bring it back. I don't even want to kind of mess with that stuff. But if you do find yourself having to tweak your compensations, again, talk to some of the experts at the core facility. They'll give you some advice about how to optimize maybe some of your controls, your stained, your unstained, and you'll never have to, fall into that pitfall of trying to move things manually. So again, that's the default of FCS Express, pulling in the compensation from the time of acquisition. Now, in this data set that I have, and let me see if I can't pull this up again, I have a number of compensation controls, right? So I've got this is like a 5-color experiment with an unstained control. It wouldn't matter how many parameters you've collected. Everything we're going to do today is going to work the same exact way, whether you have 2 parameters or you're working with a BDX50 and you've got 50 parameters, right? As long as you have these single sane controls, you're going to be all set regardless. So what I'm going to do is open up a new layout. And when you're doing post-acquisition compensation using FCS Express, you want to be using a new layout to perform your compensation, to do all the compensation setup, and then we're going to be using that to generate a compensation matrix that we're going to open up in a separate layout to actually perform our analysis. So again, what we're going to do here is we're going to use this blank layout to create our compensation matrix. Once we're done running the wizard, we're going to use that in an actual analysis template or create our own template to apply that compensation. So again, the place to start is the compensations and a mixing dialog. And we're going to come up here and say, create a new compensation. And this is our wizard, right? There's a 6-step wizard. In reality, we're going to be using just 4 out of these 6 steps. We're going to ignore these kind of optional ones for today. And what FCS Express prompts you to do is first select your single stain controls. So I'm going to hit that little load button, and I'm going to go to my desktop where I've got some data, and I'm going to grab my single stain controls. So what I can do is just multiple select all of the controls I want to load. It's 6 controls today. Again, if you have more controls, you can add them in anytime that you want. And let me change my file format here to the proper format that we want to use today. And when you do that, FCS Express is going to load in all of your controls, and it's going to match if you have labeled your controls with the name of what they are, FCS Express is going to try its best to match, say, APC to the APC parameter in the data set. Right. So if you just label these compensation control 1, 2, 3, 4, 5, and then your lab notebook, you said APC is 1, FTSE is 2, PE Sci 5 is 3, right? The matching is not going to work. We're not going to magically doing something there. We're actually reading what's in the file name when we're trying to match it. If you do need to change, right, you can pick and choose from the list. If you didn't annotate as well as you should have, you can pick and choose the parameter from the list. Now you can see that we've also automatically identified the negative control, right? So down here, the file that we loaded here was our compensation unstained control. So since it says unstained, if it said negative, something like that, FCS Express would recognize it automatically as a negative control. And then for all of the other data files that we have here, it's going to use that as the unstained control. Now, if you're working with internal negatives, right? So you've kind of mixed together negatives with one single sane control. FCS Express is going to accomplish that as well. If you have an internally sane control, FCS Express is going to recognize that, and it's going to automatically say use this as a self negative. All right. So there really isn't too much you have to do. As long as everything is kind of annotated as most people do, 95% of the time, things are going to load automatically. But if you do need to kind of force things to use an internal control, or if you have a mix of internal controls and negative, unstained controls, you can utilize that kind of mix if you want to. So the next step in our wizard is just identifying what we want to use to create a gate. So generally what we do in our compensation wizard is we try to automatically create a scatter gate for you, right? We know we want to kind of exclude things like debris, even when we're, performing some sort of compensation. So again, FCS Express is going to try and match up what it thinks you should be using for your scatter parameters. If you decide today you want to use forward scatter H, side scatter height, you can change those. And the next step here, and again, this one's going to happen pretty quickly. So what I want you to keep an eye on is when I click build histograms, down at the bottom of the page, about down at the bottom of the layout, we're going to create multiple page tabs, one page for every one of the controls that you've created. So again, I'm going to click build histograms. SCS Express is taking a look at what you decided are the single scene controls, and it's created a number of pages, one for every control that we have. So in this case, we have our APC control up here. We just kind of pick one, what seems to be representative data file to identify our scatter gate. And these gates, they work just like any other gate in FCS Express, right? If I move this gate, you can see down below, the gate below is moving as well, right? So for our stained control, this gate seems to make some sense, but for our negative unstained control, it seems to be off a little bit, right? So how do we adjust these things separately? Well, we go into the gating tab and we can say, make it a data specific gate, and then that gate will move independently. or you can use the shortcut key. If you hold down the shift button, which is what I'm doing right now, and you move that gate, the gate will move independently. All right. So if you have a mix of cells and beads, right, if you have some sort of combination where the scatters are going to be a little bit different, you can use that data specific gate to your advantage to make sure you're selecting the correct population, just like we did here, where the population shifted a little. So that's setting our scatter gates. We are going to review scatter gates on every single control for you on every page that we create. So what we do is we put in a plot here for the scatter parameters for APC stained. We show you a histogram where we're automatically setting the gate for the APC stained control, and we're automatically setting the gate for the unstained for that APC. Now, again, if you're working with internal negatives, you won't get two histograms. You'll get one histogram with your negatives over here, your positives over here, and the same markers already defined. So FCS Express does a really good job at peak fitting, trying to figure out, where those peaks are. In the latest kind of releases of the software, we've really kind of optimized that, so there's a lot less that you have to do. And really what you want to do is just kind of go through page by page, review that your scatter gates are correct, review that, you've selected the correct populations. Because, we don't work, the best for everything, right? It's just a robot trying to figure out where a peak is. So in this case, with our PE control, we probably do want to extend this gate out a little bit, right? We did good for the other four, but for this one, we want to extend that gate out and make sure we're grabbing the appropriate population. So once you've gone through, you've built your histograms, you've defined your gates, you've let FCS Express automatically pick where the positives and negatives are, and you've gone in and reviewed that to confirm it. We're then in the position to calculate our compensation matrix. So we can click on step 6, we can click calculate matrix, and you can see that FCS Express has calculated a compensation matrix for our 5-color experiment. All right. So it's pretty simple, right? The wizard, it's a wizard. It's meant to do things automatically. It's going to guide you through the process. And in this case, it's 5 colors, but if you have 10, 15, 20, 30, the process is going to work the same way. It's just more single sign controls. It's more pages that are in here, but you're kind of set at that point. So at this point, I've created the compensation. Generally, this is the point where I do a save because I want to be able to come back to this compensation sometime later. If I want to review the setup, if I want to, see what happens if I move these positive and negative gates a little bit, right? That's going to impact the way our matrix is calculated. So I'll just come up here and I'll do a save or a save as. I'm going to put it in here. I'm going to call it today's compensation. Okay. So that's our compensation layout, right? This is where we've defined the automatic compensation. But how do we get this matrix out somewhere else so we can apply it to our samples, right? At this point, we've just kind of looked at our controls to create a matrix. So what we do is we say export this compensation, right? We click this little save icon and we can save this and we can put this anywhere we want. We can call it today's compensation, right? You might have one for different days. If your instrument is pretty stable, maybe you'll use the same one a few times, right? But we have that compensation saved. We have this layout saved. So really what we can do at this point is we can close out this layout, right? We don't need that layout where we've created our compensation anymore because now we're ready to perform an analysis. So what we'll do, we'll open up a new layout. This again is where we'd probably add some data. So we'd open up the data list to kind of get ready for that. But what we want to do now is use this compensation for any data files that we bring in and we want to start analyzing. So instead of clicking the save button, right, what we're going to do now is load up a compensation. There's a little load icon here, right? And what we're going to do is go over and grab today's compensation that we set up. We're going to open it up into FCS Express. And when we expand this, we can see the compensation that we created in our separate layout. Now, if you want to use this compensation, which you probably do for all the data files that you're loading in, the easiest thing to do is to right click on this and say set it as the default. Now, if you set this as the default compensation, any data files that you load into this layout in FCS Express are automatically going to use that compensation, right? If you've been to one of our previous talks, there was a few extra steps you had to do to actually get that default to apply to all data files. We've listened to your input and we've made this a lot easier. So now if you just set that as the default, FCS Express is automatically going to use that compensation. So what I can do is I can just get this data into FCS Express at this point. I can kind of move this out of the way. I don't even have to look at it. But if I drag and drop this data file into the software, it's now using the compensation that we created. It's using this new compensation default that we had set up. And again, if we come in and start looking at, CD4 versus CD8, we can see that our data is compensated, according to the controls that we've used in this particular experiment. All right. So again, setting up these compensations, it's pretty straightforward using the wizard. I wish it was kind of more complicated. It would give me something more time to kind of talk about it. But setting it up is pretty straightforward. Once you set this as the default in FCS Express, you're able to use that. And just like I showed you to kind of swing back to the beginning, if we want to, create multiple plots, I just did a copy and paste there, and I want to format the overlays on this plot. If I want to compare compensated to uncompensated, right, here's my uncompensated because I use that formatting overlays to remove it. I put it back. I have it over here. Again, if I have compensations in here, I can drag and drop to the plots to get them to apply it. And again, it's very pretty streamlined at this point. So again, compensation is pretty darn straightforward. If you're working with a compensation from the time of acquisition, nothing you have to do. If you're working with post-acquisition compensation, you set up a layout, you create your compensation, you save that layout, you save your compensation, you load that into a new layout when you're ready to perform your analysis, set it as the default, and then we're going to be automatically using that for any of the data files that you're working with. So with that, I wanted to see if there's any sort of questions or comments that folks have about compensation before we move on to the next topic about talking about some of the high dimensional data reduction tools. Um, and just to kind of relay a message that came into the chat window, just as a reminder, using those slider bars impacts data integrity, right? If you want to have reproducibility, if you want to use controls and not your intuition to kind of move data around on plots and things like that, you've got to be doing automatic compensation, right? You got to be using these algorithms, these mathematical matrices that are set up and have been being used for many years by all software packages to do this automatically for you.
Yes, and Sean, that's great, and thank you for reiterating that point. And what I'm putting in the chat now is also a flow post-it from our lab on how you can, quantitatively approach compensation and unmixing to see if it's accurate. So if anyone wants to take a look at it, I put it in the chat. I think there was a question also from Rashana in the Q&A.
Yeah, so I saw a question here. It says, when we use this for unmixing from SciTech, do we use the files in the raw or unmixed folder after using the live unmixing? So if you're doing unmixing, and again, unmixing is something separate from compensation. We approach it the same way in FCS Express, but up here in our compensation is a unmixing navigator. But if you want to define an unmixing in FCS Express, we choose an unmixing. And you would be using the files from the raw folder. When you're doing an unmixing, you're creating an unmixing matrix, and you'd apply that to a data file that you'd want to analyze later. So when you're using SpectraFlow, you're doing unmixing using the raw folder, the raw unmixed the raw mixed data files, and then that unmixing is applied, and that's how you get those data files that come out in that unmixed folder. So I gotta say, SpectraFlow does an exceptional job at unmixing. There's nothing that we're doing in FCS Express that SpectraFlow can't do or doesn't do easily. So if you're unmixing in SpectraFlow, and when you bring in those unmixed data files into FCS Express, there's nothing you have to do. They're already unmixed, right? If you do want to do unmixing in FCS Express for some reason, you want to use different autofluorescent controls, you want to compare different kind of negatives and things like that, some permutations, it's a pretty good tool for that. And again, if you're doing unmixing here, use it from the raw folder, and then you can apply that to the raw data files that have come out of the SciTech instrument. But a mixing, it's something we'll leave for another day. It's similar, but it's not necessarily related to compensation. So great question there. All right. So with that, let's switch gears a little bit here. All right. And I'm going to kind of go back to PowerPoint a little bit. Let me get FCS Express launched up again, just so we have it ready for us when we're coming through um, all the high dimensional data reduction stuff. So give me a second here to load this up, and I'll call up my handy dandy PowerPoint here. So what I want to do today is for high dimensional data reduction is, again, give folks a little bit of a primer, right? One of the things that I find is, people go into high dimensional data reduction not really understanding what it is they need to do, right? How do they interpret t-SNE plots? How do they interpret clusters? What are the different settings used within these kind of tools and algorithms, and why do we even use them, right? So I'm going to kind of start there, right? We're going to give a little bit of an overview of how these things work, at a very high level, something meant for scientists, not bioinformaticians. I'm coming out of a scientific background, not a programming background, so I'm right there with you. So, when we work with high dimensional data, what's the problem? And the problem, it's a double-edged sword, right? It's too much data. You have these instruments now that can acquire, 20, 30, 40 parameters. So, in the end, we really just want to distill some information from all of that that can be really interpreted as some sort of biological response, right? Finding the size of a few cell populations. And the solutions that we have changed, right? In the past, what we really had to do was analyze the data by looking at 2 dimensions at a time, draw manual gates on that, and kind of using our intuition and experience as scientists to interpret what that means. And I don't mean that sarcastically, right? I mean, in the past, we had to look at a big matrix of plots like this, and we had to figure out, what's going on in, all of this data to just arrive at that kind of one rare cell population, and then compare that to other data files in some way or another. So, that's something we used to do in the past. It's something we do currently. It's not a bad solution. It's something that works. It's just very time consuming, right? The other kind of solution that you can have is do some sort of really complex math in the n-dimensional space and get out an answer and trust that answer, right? And that's what bioinformaticians do, right? They write tools, they write software packages to do really complex math that can give you an answer as a scientist, and then you have to trust that answer. And you can trust those people, right? They're very smart people doing very smart things, but that also, there's some negatives to that you have to have one of these people on your staff, right? You have to work with them all the time, and you can't really visualize what's going on with that data as easily. Now, the kind of third way, and this is where we've been kind of heading in the last few years of flow cytometry, is find out a way to visualize or figure out a way to visualize data on a single plot, or a few plots, and try and figure out how to interpret that plot. And that's what we do with tools like VisNE and Spade and FlowSom. We're taking a 40 color, just for instance, it doesn't have to be 40 color, but you take a 40 parameter data set and you put that into a t-SNE X and Y plot, and then you try and figure out, all right, what does that actually mean? So hopefully by the end of today, your mind is not going to be blown by this n by n, dimensional, visualization matrix of all this stuff, and you can use some of the tools, designed to help break down some of that data. So it's a little bit of a primer on t-SNE, right? What do t-SNE clusters mean? What are t-SNE clusters, right? So I like to show this little diagram here, right? t-SNE wasn't just designed for flow cytometry data, right? It's just a tool that we use for flow cytometry data, and you can plug anything into a t-SNE algorithm as long as you can get some information, including information about clip art images from dogs, right? And I thought this is a kind of good place to start, because this Disney algorithm was fed these images of dogs, and it was probably getting information out about the size, the shape, the colors, the pixels in these things. And the Disney algorithm kind of started segregating these little clip art pictures into clusters. So there's a little cluster of dogs with mostly white, black backgrounds, dogs with mostly black backgrounds, dogs with brown backgrounds. There's kind of colorful dogs. There's dogs over here that are kind of hand drawn looking, right? So there's a lot of different things that are going on here. But what do these clusters mean, right? The one thing that Disney did not tell us is what these should be interpreted, right? What are the phenotypes of these things, right? Are these dogs with white backgrounds, or are they dogs with black ears, right? Are these dogs that are, shadows of dogs, or just dogs that are black backgrounds? We don't know, right? We have to interpret that data as a scientist to make some sort of decision. And if we kind of take this a little bit further, right, this is more clip art pictures, but I thought this is a kind of nice way to break this down a little bit further. I'm going to zoom into this kind of intersection of these two red boxes here. And I know it's kind of hard to see on your screen unless you have a bigger screen here, but up here we have some pictures of people fishing with their kind of exceptional catches of the day. In the middle here, we have some pictures of looks like, cows and buffaloes and horses. We have pictures of jack-o'-lanterns, pictures of, what looks to be like food and pies, and pictures of mushrooms, right? So, when we look at this, we can't just assume that because there's fish here and there's horses here that this is the animals, right? We can't make that kind of leap that just because these things are close together, they're all animals, right? Because if we look here just as close, we have pumpkins, and those are certainly not an animal, but they are, kind of close in this kind of space that we're looking at. Just because we have pies here that are close to pumpkins, it doesn't mean that they're pumpkin pies, right? And because we have mushrooms that are down here close to food, it doesn't mean that they're food, right? In fact, these look pretty poisonous or hallucinogenic, right? But what we have to do as scientists is come in and look at these Disney charts and interpret them. Right? We have to say whatever is in here is fishing, whatever is in here is animals, whatever is over here is pumpkins, whatever over here is round foods, and whatever is over here is mushrooms, right? That's what we do as flow cytometrists. We identify and phenotype populations of interest. And that's what we've done. We've set gates and we've labeled what those gates are based off of what we see in these Disney plots. Now, when we start working with flow data, the same thing applies, but we're just not looking at clip art pictures of dogs or things from the internet, right? Generally, when we look at a Disney chart like the ones down here, we generally display these as a density plot or a contour plot with some sort of heat assigned. And that heat, the amount of redness in these plots over here as opposed to the amount of blueness, represents the intensity of some sort of marker on that plot. And by comparing multiple plots showing heat for different markers, we can start determining phenotypes. We can set gates on that and start labeling it and start defining what those are. So here I've got a Disney plot. I've got three of them, and I'm showing the heat of CD3 on the left, CD4 in the middle, CD8 on the right. Right. So if I need to come in here and set a gate that's CD3 positive, CD8 positives, well, I can select this population because it's hot for both on both of these graphs. If I need to set up a phenotype for CD3, CD4s, right? It's hot for CD3, it's hot for CD4. So there's my phenotype, right? If I wanted to take this a step further and identify kind of three phenotypes, this one's CD3 positive, CD4 negative, CD8 negative. And that's what we're going to be doing as scientists with these Disney charts, these VISNE charts, is that we're going to be creating gates on them, trying to identify what these populations are. And then once we've done that, then we can compare between different data files, right? We can take our treated and our untreated, our control and our experimental, and we can look at the differences between the heat and the distribution of these populations to get some sort of scientific result. Now, when we look at Disney charts, another kind of a pitfall that folks run into is I ask folks here, what's the difference between the top row where I'm looking at, Disney chart with CD3, CD4, and CD8. Same thing down here, the populations look a little different, the clusters look a little different, right? Normally I'd ask everybody to raise their hands and give me your best input, but I'm just going to tell you today. So in the top row and the bottom row, we've used the same Tisney algorithm, but we've told it to start in a different place, right? So if you think about what an algorithm does, it does a mathematical calculation starting somewhere. Right. And that could be when you run a cytometer, it could be event number one that goes past the laser. It can be event number 5,000 that goes past the laser. Right. So in the top plot, we told it to start at, say event number one. At the bottom, we told it to start at event number two. So our distributions are different, right? Or if we had different data files, if they have different number of events and different, events with different values, the distributions will look different where you start or, what kind of data file you start on. Because these are different, though, it doesn't mean that the clustering information isn't the same, right? I ran the same data file, um, but it's showing here that the CD3 positivity, this little cluster that's up here, it's probably the same one as here, not just because it's the same shape, it's kind of the same size. We have a bigger kind of cluster over here. It's probably the same one there if I went in and set some gates. So they've moved all over in this plot, but it's really the same clusters. It's the same phenotypes, just existing in different positions on the plot. So, because of that, one kind of pitfall that folks run into is they think, all right, well, because something on this plot is further away than something else on this plot, that they're very different. Right. And that's not true because something has a global, a kind of distribution on this plot that's separated from another cluster on this plot. It doesn't mean that these are phenotypically further apart. Right. Just because this is close to here, it doesn't mean that these two clusters, whatever it is that they are similar to each other. It's just that in this space, they've, Tisney found a way to kind of stretch this data apart and visualize it and it put them close to each other. So, two things. Tisney is randomly initialized, right? Just choosing where you start can change the shape, size, and location of a cluster, right? When we run Tisney in FCS Express, we always start in the same position. I'll show you how we change that later. But importantly, the kind of global, size and locations of clusters has no meaning with Tisney. So, I can't tell you how many times I've gone to a conference and a bunch of very smart people are looking at a poster and I hear them saying, "Oh, look how close that Tisney cluster is to the other one. That must have some sort of, relationship." It doesn't, right? So, keep that in mind. So, I kind of summarize those 2 points. And again, it's up to the researcher to identify and determine what those clusters are. Now, there's kind of tricks to helping algorithms run more quickly. And, when I talk about differences between Tisney and Visney, one of the tricks is kind of, approximating or making estimates about your data. So if you're running a Tisney on a million event data file, that can take a really long time, right? The algorithm is actually very computationally heavy. And then you hear people talk about Tisney and Visney and what the differences are. Well, Visney kind of makes a bunch of estimations about your data. And that way, we don't have to run, say a million data points. We still kind of run a million data points, but Visney is kind of a way to make that algorithm run more quickly, making certain assumptions about the data. And another way that we do that is through upsampling and downsampling. So you may have heard people talk about upsampling and downsampling. You may have not, but I want you to kind of understand this because it's really important when we run Tisney. So what downsampling is say we have a million events that we want to run an algorithm on. And we know that a million events in that algorithm is going to take us 3 days to run, right? It doesn't, depending on your computer, I'm not saying FCS Express takes 3 days to run any of these. It's actually very, very fast. But if you could run 100,000 of those events and get the same results or similar or close enough results, and then have that done in, maybe an hour, well, it might not be worth 3 days time running a million events when you can run 100,000 and get the same result. Right. So downsampling is just a way of reducing the number of data points so that you can run your algorithm more efficiently. But the trick is to keep as few data points as possible without losing the overall structure of the data, right? So if we have a million data points, we can't run a Tisney on one event or 10 events and expect to get some sort of meaningful event, meaningful result. We have to kind of find something that's going to work nicely. Now, if you think about, all right, we have a million events and we're going to take 100,000 of them. Well, there's a lot of ways we could kind of cut that, right? We could do simple random sampling where we say, just grab event number 1755, 2786, and, do that across a million events. We just randomly grab data points that are in there. Or we could be a little bit more structured. We could say, take event number 1, 5, 10, 15, 20, every fifth event up to a million, and that will give us 200,000 events when we're done, right? But the problem is with biological data, and when we run flow cytometry, it's actually kind of an interesting problem. Biological data is unique because generally, often those really rare populations, things like stem cell populations, minimal residual disease, things like that. That's the most interesting stuff for us as scientists. We want to know what's going on with that 0.1% of data files, right? So if you think about it, if you had a full data set and you just randomly sampled 1,000 data points, well, if I have this kind of rare, kind of light blue stuff up here, if I grab 1,000 kind of random points from there, eventually I'm going to sample that stuff out, right? If I keep taking, just randomly grabbing data from there, my 0.1%, eventually I'm going to run into 0% of cells. And then my algorithm is actually not giving me anything meaningful because we've thrown away all of that data, right? So another way of kind of downsampling data is called density dependent downsampling. And it's something that's classically been done with the SPADE algorithm. FCS Express has actually made it possible to do density dependent downsampling with TSNE and VISNE. So it's kind of unique thing to FCS Express. But what we'll do is just grab more points from less dense areas. So that way you're not just, kind of filtering out all the really rare, cool stuff you want to look at and grabbing fewer points from very dense areas. And so density dependent downsampling, it's a long way of saying grab more from less and less from more, right? It's really straightforward. So say we grabbed the million events and we downsampled that to 100,000. Well, what do we do with the other 900,000? Right? We've got all that data. Do we really just get rid of it? Well, the answer is no. In FCS Express, we can upsample that data. So we can say for the 100,000 data points we've actually looked at, what's going on with the other 900,000, right? What we can do is assign some sort of best guess about where that data is going to fall and make some assumptions to kind of apply that back to the rest of the data. And again, that's another unique tool in FCS Express is we can do that upsampling portion. All right. Now, the one more thing before we kind of jump into actually looking at the tools in FCS Express, and I know we've got 10 minutes left here, and that's by design, right? Working with the tools and the software is actually pretty easily. But you have to compare apples to apples. And like I said in the beginning, we oftentimes get messages from customers or folks that are just kind of starting with this stuff. And they say, I've got 100 data files and I ran TSNE on all of them separately. My TSNE plots don't look the same. Right? It's because you have to merge all of that data together before you run a TSNE algorithm, right? You can't just take one data file, run a TSNE, apply that to another data file and expect the plots to look the same. You have to merge all of that data together. You have to run the algorithm. And then after you get your results, you have to kind of deconvolve that data to all the individual data files. So if you have, data file 1, data file 2, you ran the TSNE separately, you're comparing apples to oranges. If you merge them all together, run the TSNE, deconvolve them to look at the data files, you're comparing apples to apples, and that's the correct way to do it. So, you might say, all right, well, I've got 100 data files. They're all a million data points, right? And I got 100 million data files or 100 million data points. Isn't that going to take a long time to run and analyze? And the answer is yes, it is. The more data you give these things, the longer it's going to take to run. And I know it seems like a kind of surprising thing sometimes to hear that, but that's what these labs that are doing, big TSNE runs on, and they're comparing, 5 controls to 100 patient samples. They're merging all of that data before they actually do it. So you might be wondering, all right, well, it seems hard. You got to merge all of the data. You got to do all of this upsampling and downsampling. Why even do it? Well, one is kind of gate reduction, right? That's kind of one reason. If you, instead of creating, 10 different levels of a hierarchy for gating, if you can look at a combination of maybe, 7, 8, 9 TSNE plots, and you can identify the populations of interest, well, you don't have to create gates through many, many different parameters. You can just use the TSNE X and Y to do that. You can also use Tisney plots to try and find, differences between responders and non-responders, trying to find differences between rare populations or hard to gate phenotypes. But the other thing I see a lot of people doing is because it looks cool, right? You're going to a conference and your PI says, "Oh man, everybody's using Tisney plots these days. We better use a Tisney plot, otherwise we're going to be left behind." Well, we do see a lot of that, right? So make sure you're using Tisney for the right reasons. Don't just use it because it looks cool. The plots do look pretty sharp and nice, especially when they come out of FCS Express. But if you can get to that answer more straightforward by doing 2x2 plots and doing kind of standard flow cytometry, so to say, don't kill yourself about working with the Tisney algorithm just to try and get a Tisney plot on your next poster or the publication. But if you insist on it, of course the tools are easy, and we're going to show you how those look. So let's go to some data. Again, we're just going to spend a few minutes actually running Tisney, because Tisney is actually that easy to use in FCS Express. But again, I wanted to kind of share those kind of common pitfalls that people run into. So when we get started analyzing some data in FCS Express, again, one of the places to start, you want to get some data loaded up into your layout. So I'm going to grab some data I want to run Tisney on. And I've got 3 files I want to compare. And if you remember that last thing I told you, the first thing you have to do is merge data files. So when we merge data files in FCS Express, we choose merge FCS file from the files of type. We multiple select the data files we want to merge together, and we say open. And that's going to bring those 3 data files into our data lists. We can drag them and drop them onto the layout. We can put in whatever plot that we want. And this time parameter looks a little funky because we've got 3 different data files with different time. But what we can do, just like normal, if I want to look at forward scatter versus side scatter, I want to create some sort of gate on here, right? And my gate window is on the other screen, right? I can drag this out. What I can do now is, if I want to kind of deconvolve and start looking at individual data files, there's something here called a classification identifier, right? And you can see here what it's going to do. It's actually going to show me what those 3 individual data files look like across CD3, right? So this is another good way of, other software calls this concatenation. If you want to visualize data for antibody titrations and things like that, we make it pretty simple in FCS Express to do it. In the latest release of FCS Express that came out just last week, you're going to have this new classification identifier on these 2D plots, right? So if you're working with an older version, make sure you get updated so you can make the best use of that. Now, what we want to do for this data is run a transformation, right? In the Tools tab, Transformations, we want to run a Tisney. So in order to set up a Tisney, we choose a Tisney. And, when we run a Tisney, the less events we can throw at this thing, the better, because it is computationally intensive. So, instead of running it all my beads and debris and things like that, I probably come in here and say, all right, well, use the scatter gate, right? That's one way to downsample data, right? When I say downsampling, we're only sampling 73% of our data because the rest is debris or beads, all right? So when we run this transformation, we have to choose a few things, which parameters we want to include in the clustering. So I'm going to come in here and pick and choose what I want to include in the clustering. And down here, we have a number of kind of variables that you can set. I'm not going to go through all the details of what these variables are today, but rest assured, we have full documentation. We have a lot of resources to help you out, figure out what these things do. But the only thing I'm going to do here, in addition to the defaults, is say use OPSNI, which is a way to kind of speed up Tisney algorithm. And I'm going to say estimate Tisney for unsampled events. And that means that we're going to sample 3,000 events. We're going to downsample this data to 3,000. And for all the other events in that data file, we're going to try and estimate the Tisney X and Y coordinates for that. All right. And now to get this Tisney transformation applied to a data file or to a plot, all I do is I drag and drop it to a plot. And when this plot says Tisney transformed in the title, or once this little kind of blue border goes away here, that's how long it took to run our Tisney. And now we have new Tisney Y and Tisney X parameters. And this is my Tisney plot. All right. So again, I think this is very easy. The interface for actually accessing these tools is very straightforward. It's very streamlined. I think anybody can come in here and kind of do this, right? Now the important part is trying to figure out how to interpret and set up these plots to do some analysis. So, as I mentioned, all Tisney plots, or a lot of, times when people are looking at Tisney, they have the heat on these plots based off of some sort of parameter. So our default with density plots in FCS Express is to show the heat of counts, right? How many, how dense is that particular area for a particular cell line. So what I'm going to do is change that by showing the legend on this plot. I'm going to put the legends just below it. And you can see that it says count. But if I change this to CD3, well, lo and behold, right, there's our CD3 positives. If I change this to CD56, or if I change this to CD19 or CD4, we're able to visualize the heat of all of those different parameters. Now, we do know you have a bunch of parameters to look at. So generally what folks are doing is once you create one Tisney chart that you like the display and the formatting on, you can say color by all parameters, and you can pick and choose which parameters you want to show in the Tisney plot. And FCS Express will create a group of plots that show the heat across different parameters. And just like that first density plot, I do want to pop up the legend here and say put the legend on the bottom. And that way I can come in and say, all right, well, here's the heat of CD3, 56, 45, 4, 19.8. And now I can start creating gates on these things and start, analyzing what's going on. But if you remember, what I mentioned is, if I have 3 data files, well, I have to kind of deconvolve this data in some way or another, right? So I have to go back and have a way to go in and say, all right, well, this is file 1, this is file 2, and this is file 3. So we use gates to do that, right? So if I come onto this 2D plot and I say, this is sample 1, I create a gate for sample 1. And what I can do is I can grab that group of plots and I can say apply sample 1, right? So what I'm doing now is I'm viewing in these plots what falls within that gate. And if I want to move the sample 2 or to sample 3, I can adjust the gate. Or if I want to group sample 1 and sample 2 together, right, I can do that. If I want to create, another gate on here, I'll call this sample 2, right? I can create another gate. And what I can do is just simply duplicate this group of plots. If I really want to kind of compare them side by side, well, I can take that second group of plots and apply the sample 2 gate to it. And that way I'm comparing sample 1 to sample 2. So, that's how we arrive at some of the plots and the displays for working with Tisney and FCS Express, right? It's very straightforward. It's going to be here in our tools and our transformations, right? There's no R plugins, there's no programming, there's nothing you have to do to kind of access that. But there's also lots of other tools and algorithms out there that are out there on the market. Things like UMAP and FlowSum and FlowAI. There's new kind of acronyms for these things almost on a monthly basis these days. So what we've done in FCS Express, and let me do this in a new layout so we don't get kind of too confused, and I can come back here and take some questions in a little bit for Tisney as well. What we've done in FCS Express is we've created something in the software. Again, this came out just last week. It's called pipelines, okay? Because all of the algorithms that most people are working with on the market are just some sort of kind of iterations of each other. There's different steps that go into these things, right? You never see what they are, right? There are programming steps, there are scripting steps, they're kind of all these things that go in. But what we've done in FCS Express now is we've enabled in the transformations something called pipelines. And what a pipeline will do is they'll allow you to grab, whatever parameters you have and apply different steps to this data. So if we wanted to downsample this data, right, and just take, say 1,000 events or, 2,000 events or something like that, we could put in a downsampling step, right? And again, that's a step that's also included in something like Tisney. And let me change this to a color dot plot. We'll look a little bit nicer in a moment. Now, if I want to do UMAP, right, that's another thing we've heard a lot about lately. Well, I can put in a UMAP step and I can make sure I get these in the correct order. And I can say downsample that data to 2,000 events. I can say run UMAP on these particular parameters. that are there. I have, all the variables for UMAP, and I can say apply that pipeline to the plot, just like I would with any transformation or compensation. And you can say it says pipeline transformed. So now I have UMAP 1 versus UMAP 2, and I can start working with these UMAP clusterings to do, exactly what I showed you before with density plots, using the same type of things for TISNE. Now, we know that you don't always know, what all the component steps are in all these algorithms, right? UMAP's pretty straightforward, but if you're working with something like FlowSum, right? We have FlowSum here in our predefined algorithms because FlowSum is a number of steps that are kind of put together. There's a scaling step. You have to create a self-organizing map. You have to create a minimum spanning tree. You have to create a graph layout. You have to create a consensus clustering for meta clustering. And then at the end, you might even want to remove some of those scaled parameters. Now, those are all the things that go on behind the scenes when you use something like FlowSum in any software. But what we do is we just group all of these together for you. And that way you can use them right off the bat, right? Maybe you did a FlowSum and you did meta clustering, but then you want to take those meta clusterings and then do a UMAP on those, right? Well, what you can do is you can add UMAP right to the end there. And then when you run this pipeline, you can run a FlowSum and then meta cluster using UMAP, right? If you wanted to do, some other type of downsampling to this data, you can pick and choose what type of downsampling you want. You can arrange it into that pipeline, and you can randomly sample that data any way that you want. So what we're hoping for is that the pipelines in FCS Express are going to kind of, make working with these algorithms more straightforward and easy to use, really for everybody that's out there, not just people that are here, people that know how to program these things, that know how to use plugins, that know how to use R, right? We're trying to make that available to everybody. And I'm expecting we're going to see some really cool new combinations of people kind of coming up with their own pipelines, their own algorithms for running these things outside of, what you can just get from R repositories and GitHub and, programmers and things like that. So with that, I just wanted to kind of wrap up and say thank you for joining us today. One thing I have up here on the screen is our support email, support@DeNovoSoftware.com. So if you have any questions after the call today, or if you're watching the recording of this, make sure you reach out to our support team. Everybody in our company gets those messages, and the person kind of best positioned to respond to you, whether that be through their expertise or their geographical location, will generally get back to you within an hour or two, never more than 24 hours. So with that, I'll kind of open up the floor here. If folks have any questions or comments, you can type them in the chat window. You can type them in the Q&A window. I'm happy to take any of those as they come in. And if not, again, thank you very much for coming out today.
Thank you so much, Sean. It looks like we don't have any more questions coming through. So it looks like we're good to go, and we'll hopefully see everyone next week for the session on the 9th, which is getting started in FCS Express for previous FlowJo users. Okay.
All right. Thanks again, Kathy. Thanks, everybody. Have a good day.
All right. Thanks, Sean.